Okay, today I'll show you how to design a pair of cloning PCR primers to clone your favorite gene. Also, we can call them the gene of interest. So the gene of interest can come from any species. For example, let's say this is a plant DNA and the red box shows a gene that you are interested to clone, and the arrows pointing towards each other, we are calling these arrows as forward primer and reverse primer. And the forward primer is pointing from 5' to 3' orientation, and reverse primer also is pointing from 5' to 3' orientation, but it's in the opposite direction. Now the nomenclature of forward and reverse primers are subjective, and it's just we name it for our convenience. So now this gene that's present in the plant chromosome, okay, and basically your goal is to clone this gene and clone it into the, so this is your gene of interest, you want to clone it and you want to insert it into your plasmid DNA, okay. So now before you do that, let's say that you want to amplify this gene of interest from your plant DNA, and then you want to attach two sites, two restriction cut sites. So namely, let's say you want to attach a BAMH1 cut site on this side of your primer, and you want to attach an EcoR1 cut site on this side of your primer. Why you want to do that? Because your goal is, after you do the PCR amplification, you want a BAMH1 cut site hanging on this end of the forward primer and an EcoR1 cut site hanging onto the other end of the reverse primer. Now if you are asking that why did I choose these two random enzymes, BAMH1 and EcoR1, my answer would be because this is the plasmid that you have in your lab, right? And this plasmid has a cloning site that was digested or cut open with BAMH1 and EcoR1. Or in other words, the manufacturer of this plasmid or the company or the person or the individual who synthesized this plasmid designed and synthesized in such a way that it has this BAMH1 cut site and EcoR1 cut site. Now remember, the plasmid that you'll be using in your lab could have completely different cut sites. And if you know what is a multiple cloning site, then sometimes in the multiple cloning sites, you might have 3 enzymes here and another 3 enzymes are here. Okay, so you can choose any of those 3 enzymes here, one of those 3 enzymes here, and one of those other 3 enzymes here. But every time you choose an enzyme, you have to be super careful so that you do not have a restriction enzyme, same restriction enzyme cut site here. So just imagine what's going to happen. Let's say that you want to use BAMH1 cut site, and unfortunately your gene of interest also has a BAMH1 cut site, okay, and you didn't know that. So you attach this BAMH1 cut site here, but since you did not know that your gene of interest has a BAMH1 cut site, your BAMH1 enzyme, when you will be using it to clone, will not only cut here, it's going to cut here too. So that will be really counterproductive. Now you are truncating your gene of interest, so which is really bad. I'm going to show you at the end of the discussion how you can be 100% confident that this gene of interest does not have any of these cut sites. I mean, neither BAMH1 nor EcoR1. Okay, it's very important that your GOI does not have any of these cut sites. Anyway, so your goal is to clone this gene, okay, and you want to insert your gene into this plasmid vector. In order for you to clone this gene of interest, you need to first amplify it using PCR. So you need to design a pair of primers, and then we attach this BAMH1 cut site and EcoR1 cut site. Okay, So let's go back to our favorite gene bank, and let's say that this is a chlorophyll synthase gene, and I showed you before that how you can identify this gene from gene bank through a simple gene bank search. It came from the cotton plant, Gossypium hirsutum, and as you know that if you click the CDS, the coding sequence, it's going to show you only the coding sequence of the gene. If I click the FASTA file, so now the FASTA click, if the FASTA format, then all the line numbers gets deleted, and here is your gene of interest. Now you need to amplify this gene of interest. That means you need to design a primer. So forward primer will be from this side, it will anneal to this end of the gene, and the reverse primer, it will anneal to the other side of the gene. Okay. However, when you design the reverse primer, you have to be super careful, because the reverse primer, you have to first identify the reverse complement in order for you to do the reverse primer. A lot of students make a mistake, and I'll explain to you how do you identify the reverse complement. Okay, so now what you can do is, let's copy this entire gene sequence that you want to amplify, and then I'm going to go back to the slide presentation. And this is my gene sequence. Okay, so first what I'm going to do is, now remember, by default anytime you copy and paste a gene sequence, this is going from 5' to 3' orientation. Okay, so it's going from 5' to 3' orientation. What do I mean by that is, so this is your 5' and this is your 3'. So by default, it is moving from 5' to 3' orientation. Okay, so let's first choose, let's first design the forward primer. So I'm going to take about 20 bases. So 1, 2, 3, 4, 5, 6, 7, 8. Okay, so from the forward, for the forward primer, I selected the first 20 bases. So let me copy it. Okay, it's very important that you write 5' here, and then copy the primer sequence, and then write 3' on the other end. Okay, so it's very important that you write 5' and 3' end, because you remember when you send this sequence to a company to synthesize your primer, they will always assume that this is a sequence that you gave 5' to 3' orientation. If you make a mistake, then you will not be able to do your PCR. So it's very important that you give them the 5' to 3' orientation primer. Okay, so that's my primer for forward primer, but we are not done yet. Why? Because remember, according to our plan, I need to insert a BAM-H1 cut site. Okay, so in order for me to do a BAM-H1 cut site, I'm just going to, I can do a very simple, I can go to NEB's web page. Okay, and I can go to New England Biolab's web page, and then I can search my enzyme here. So you can go to neb.com, and you can also actually do a simple Google search, but I like to go to the web page to identify the primers. So I typed BAM-H1, and when I typed BAM-H1, this is the primer, sorry, this is the restriction enzyme. So this is the sequence. Notice something. So this is the sequence that I need to attach. So this is a sequence I need to attach to the forward primer. So the sequence of BAM-H1 is GGA-TCC. So I'm going to write here GGA-TCC. I'm just going to use a different color so that I understand. I'm just going to use blue. Okay, so now I attached a BAMH1 cut site at the forward primer, but we are not done yet. So it's important that you add a junk sequence before the BAMH1 cut site. Most people use the sequence GAGA. You can use other sequences too, but I like to use the GAGA sequence. This is a junk sequence is important so that the restriction enzyme can sit here and cut here. Okay. So remember, this is a junk sequence. Then this is your restriction enzyme cut site sequence, and this is the first 20 bases of your gene. Now let's design the reverse primer. Okay, so for the reverse primer, I'm going to again write 5 prime, and I will not copy and paste this sequence. Okay, and here is why. Remember, reverse primer will so this is your DNA. Okay, so 5 prime to 3 prime and 5 prime to 3 prime. So remember, at that stage of denaturation stage, your DNA becomes, from double stranded it becomes single stranded. Initially you use denature your DNA during PCR at around 95 degrees centigrade. So now what happens is that the forward primer that we designed, remember we designed just forward primer, it binds to this strand. Okay, and our forward primer we wrote it was in the 5 prime to 3 prime orientation. Okay, now the reverse primer will attach to this strand. Okay, and in this orientation of 5 prime to 3 prime. Okay, so that's your reverse primer. Now if there is an A here, okay, your reverse primer will have T. Now if you have a C here, the reverse primer will have G. If you have C here, the reverse primer will have another G, and so on. So basically the reverse primer, if you read from 5 prime to 3 prime orientation, it is T, G, G, and so on, so which we call as reverse complement. Okay, so when we read the reverse primer, as I showed you, you need to use a reverse complement. So that means it is going to go from this direction to this direction. Okay, so do not copy and paste this sequence like that. If you do that, for what I'm doing right now, okay, this is wrong. This is wrong sequence. You need to have reverse complement. So in order for you to do the reverse complement, what you can do is you can go to a website, like you can just go to Google reverse complement, and when you Google reverse complement, there are many websites, and the first websites I found, I just want to copy and paste my sequence, and then I'm going to submit it. So this was my last 20 bases of my gene sequence. So I'm copying it, going to reverse complement, and then I'm going to submit it. So this is my reverse complement. Okay, so reverse complement means that I am now reading from this direction to this direction. Okay, so that means I am now reading from this direction to this direction. Okay, and that is also in this direction, and that is also in a reverse complement fashion. What do I mean by that? I mean that you are reading from this direction to this direction. So A becomes T, G becomes C, this T becomes A, this T becomes another A, this A becomes another T, and so on. Okay, you can really manually do it. You can just read it backwards in this direction with a complementary strand later. So you can read, like, instead of A, you can read T, C, A, T, G, C, etc. or you can go to the reverse complement website and then find out the reverse complement sequence. Okay, now remember that we said that we want to include an eco R1 cut site. So I can again go back to the New England Biolabs web page and type eco R1 and here is your eco R1 enzyme. It is GAATTC. Okay, so I'm going to go back here and then I'm going to write GAATTC. Okay, and I'll just change the color again so that we understand. GAATTC in blue. So that's our restriction enzyme cut site. Don't forget to add the junk sequence GAGA and don't forget to write 3' to 5'. Okay, and let's change the color here again to red. So now we have a forward primer and reverse primer. Okay, so this is the way you design a forward and reverse cloning primer. Now cloning primer means this primer will amplify the entire gene so that you can clone it. So this primer will amplify the entire gene and then it's going to attach this BAMH1 cut site and the EcoR1 cut site. So once you amplify it, okay, you will get a band in the gel and you purify the gel and then you digest it with BAMH1 and EcoR1s. Then you make them sticky, and once you make them sticky, you digest the plasmid with BAMH1 and EcoR1. Then you make them sticky. Then you take this gene of interest with your sticky end and you add ligase, the enzyme that will glue this gene of interest back into your plasmid backbone. Now, I told you that I'm going to show you how you will know whether your gene of interest has any restriction enzyme cut sites, because that will be counterproductive. Okay, so I can always go to the same website that I went, neb.com, and I can search for a tool known as neb cutter. It's really interesting. So neb cutter, what it does, it's basically a tool where if you copy and paste your gene sequence to neb cutter and then just click submit. So now it did virtually cut our gene of interest into with multiple restriction enzymes, a lot of them. Now remember we have used, now you can just look at the enzymes and see whether our restriction enzymes that we have used have any cuts, whether do they have any cut sites here or not. So the easiest way to do for it, remember we use BAMH1 and EcoR1. Anyway, so to go to the zero cutters list, if you don't find your enzyme, then you are good to go. In our case, we did choose BAMH1, but I don't see it in this list. So in our case, we should go back here and do not use BAMH1 here, okay? Because there is a cut site here somewhere according to this website, okay? And let's see EcoR1. EcoR1 in is this list. That's a good news. Remember, these enzymes that don't cut. So that means we can use EcoR1 here. Okay. So in this case, we cannot use BAMH1 for our gene of interest because we might have a cut site here. We can use EcoR1 for this side for our cloning because our gene of interest does not have a EcoR1 cut site. So in your case, whenever you are designing a pair of PCR primer, be sure after you do the primer design, copy and paste this sequence to the neb cutter website, NEBCUTTR neb cutter, and you can find the neb cutter to New England Biolabs website. You can go to neb.com to find the neb cutter. You can also Google neb cutter, NEBCUTTER. So this is the way you design a pair of cloning primer for PCR. Okay, you might have a question that, okay, so I have a BAMH1 cut site in my GOIs, what do I do? Well, highly likely that this site will have, this side of the plasmid DNA will have another cut site besides BAMH1. So you'll be lucky if and I'm sure that you will find another cut site. What if you have 3 cut sites here and your GOI also have all of these cut sites. So that's really a bad luck. Then you cannot clone your gene of interest into this plasmid DNA.